I today represent the work from the National Center for Neurology and Emerging Diseases. Professor Sonia Marshall-Gradzik would like to be here with you today and also during this week, but unfortunately she has some issues with family and she's not allowed to attend the conference. So I hope I can represent well my team. And it's an honor to be here. It was one of the best conferences I have been attending. So first I would like to thank you all the participants we have involved in these researches because of course we just can grow evidences if we have the participation of them. So thanks so much. And of course we need the fundings to support our research. So I would like to extend our gratitude for these fundings. I have this slide here that of course I'm not going through much because we have experts, we have community of here, so it's not necessary, but I would like, I would like to emphasize this point here, that is the onset of exposure many patients with ME have after having biological, chemical, or also physical stimuli. And when we talk about the overlap with long COVID, these patients also have developed the symptoms after infection. So it's strongly related with TRP channels. Oh, okay. Sorry, let me try this. Okay, with TRP IO channels. And, sorry, this one working? Yeah, okay. I think this will be easy because I'm not really seeing my slides. So when we look at IO channels, we have an important response of these IO channels when they are vulnerable and stimulated by different stimulus like vibration, temperature, and here, the most important for our research, the infections. And these IO channels, they work like, if someone not familiarized with this, they are like a house is a cell, and we have doors. And these doors are the IO channel. We need these doors open to allow the important ions, such as calcium, entering the cells. And these TRP channels, they are important regulator of calcium signaling. Why it's important? Because calcium is engaged in different pathways, in different signals. It's important since for basic cell divisions, as also for mitochondrial functions. So if we have this dysregulation in TRP channels, we also have a decrease in calcium inside the cells. TRP channels have been also strongly studied in the last decade as potential targets for therapeutics. And the last point is the similarity we have with the location of these IO channels, because we have these many IO channels expressed in different parts of our bodies. What, as you know, long COVID and ME patients also have multiple symptoms. So we can relate, for example, in the neural system, we have many of these TRP channels, and we have many cognition, memory, and other issues in these patients. So just briefly about the background in our research, why the natural killer cells and why these TRP channels. Around 10 years ago, Professor Sonia Marshall-Gradzik published a paper about the SIMS from TRPN3 IO channels genes was dysregulating MCFS patients. She studied many different SNPs, and she found these 4 ones in MCFS patients, that TRPN4, C2, M3, and M8. M3 was the most prevalent one. So Professor Sonia and the group went for look other things about this TRP channel, and we identified that the protein expression of TRPN3 is dysregulated, is inferior in cells from MCFS patients. So after that, we start to look about the calcium, as I mentioned, the importance of calcium. So we test if the calcium really was reduced in the cells of these patients. And yes, there is a significant reduction in calcium mobilization in natural killer cells from ME patients. And as other groups already studied, such as Dr. Nancy, our group, and we find that there is a significant dysfunction in natural killer cell cytotoxicity. Natural killer cells are cells that have the main target of kill cells that are not working properly, like cancer cells, infected cells. So this function is not working properly, probably because we have this dysregulation in calcium, and so if calcium is not enough, no function enough. I work specifically with the technique named patch clamp. This is a technique to measure the function of the ion channel. Okay, this is the gold standard, is the best technique we have now for study ion channels, and this technique measures this current when the cells in the microscope. We have a specific setup, and it's a bit similar IVF. We have this glass pipette. We prepare cells from the patients, natural killer cells here, and we have to approach here the cell. When I get this approach of the cells, I need to open the plasma membrane to get the whole cell. This way, I can measure all the changes happening in the cell. So for know how it's trypan-3, I use specific drugs. One, that is the agonist. I use pregnenolone sulfate. That's the basic one and the best. That is working for open the ion channel. So I test if the ion channel is open to allow the calcium inside. And I use another drug that the name is ononetine. That is the opposite, is an antagonist. So I can see if this ion channel door can close properly. Here I have the first part of these data. I will present just the part of progress today because the time is short, but this paper is already published, so you can have access to the others, and I'm happy to talk with anyone interested during the lunch or after. So when we did the first paper was looking health controls and ME patients, and there is an important and significant difference in the functions of trypan-3. What means trypan-3 is not working? The door is not open and closed properly, so we have less calcium inside of natural killer cells from MCFS patient. When this study was published before COVID-19, so when we start to have the overlapping clinical presentation with long COVID, the group went for this part that was compare health controls with long COVID and ME. And if you see here, we have a significant difference in trypan-3 in the cells. However, the important data here I'd like to highlight is the no difference when I compare long COVID and MCFS trypan-3. So the dysfunction demonstrates this important overlap in both conditions. So we have great talks, I'm happy with this, about the LDN today. So everyone is familiarized with those and everything. So we look for naltrexone. And I just would like first to explain another mechanism naltrexone work that's more related to trypan-3. As you know, naltrexone is a mu-opioid antagonist. And what's important is that the mu-opioid receptor have a potential to inhibit trypan-3. So in the moment we have this antagonist drug alleviate the results, the effect of opioid receptor, we can alleviate the effect of mu-opioid receptor having blocking trypan-3, and with this we can increase the calcium. This was our hypothesis, and we start investigating in vitro. So when we select the cells from MCFS patients and health controls, incubating the same treatment and with naltrexone and compare, we have no difference. The ion channel was working the same way for these MCFS patients after treatment. So we did the same after with long COVID, and we confirmed that in long COVID, it's also working after naltrexone incubation. What reinforced the potential overlap in the mechanism that is causing the dysfunction in trypan-3. Of course, in vitro studies is really important, but we have to go for in vivo because it's much complex. We can have different metabolism that will, in a person, not work. So we repeat the experiment, but this time selecting healthy ones and MCFS patients that having taken LDN. So this patient was on treatment because the physician prescribed off-label the LDN, and we select and analyze. And yes, our in vitro results was also confirmed in vivo. These patients have the same trypan-3, what due then to have the restoration of calcium inside the cell. And when we did the last study, this study was published 1 or 2 weeks ago. We look long COVID, healthy ones, and patients long COVID taking LDN. So we reproduce our first data that we have a significant difference in long COVID and health. And again, we confirm that low dose of naltrexone work to restore TRPM3. These studies we analyze symptoms, we analyze quality of life and disability. However, we have a small number of patients because in patch clumping we focus on number of cells we are working, individual cells. So this work led us to a clinical trial. So now we are doing a double-blind clinical trial to check how it will work. And of course, here we can have placebo effects when we analyze symptoms. Other data I'd like to present is not part of the research I'm directly involved, is a research from Dr. Natalie Etianne. She is looking the immune exhaustion of long COVID in ME patients. So NCND is looking to investigate the transcriptome changes. So we are currently using the nanostring technology. We use RNA to analyze the expression of exhaustion genes. This panel we select use more than 700 different genes. And with this technique, we decide to use it because the immune exhaustion, as you know, it's really related to the symptoms we have in both conditions because it is prolonged response we have after the infection. So some data we have here today. First, about the MCFS patient, Natalie identified 14 genes that were significantly different in ME patients. So, for example, we have some genes down-regulated here in this region, and we have the upper-regulated here. So if you look, we have, for example, interferon here. We have some immunoglobulins here that was down-regulating this patient. And we also look for long COVID patients. The number of genes involved and altered was superior, 29 genes, with almost half down-regulated and other half upper-regulated. We also have genes involved in interleukin production and secretion. We have HLA genes here. And I would like you to have a look about this other overlap you have here. From the half expression genes that were altered in this study, half of them are also different in long COVID. So it's another overlap we have. But of course, this is a research we are starting. We have to look why we have this big difference too, with other genes more altered in long COVID than in ME patients. NCND is also focusing neuroimage. So we have some studies conducted with the 7T MRI. It's not an MRI machine used for clinical. It's a specific MRI machine that have a better resolution and also can see clear and better details, especially when we are looking for small parts of the nervous system. So a PhD student, Myra Inderi, she did an interesting find just published last year, looking at the significantly weaker function connection between two important parts of the nervous system, the brainstem and cerebellum. This was specific for MCFS patients, but she's currently doing the other part with long COVID, and soon it will be published. Other part our team is looking is about the hippocampus part, and I just would like to briefly tell about some points really important about the hippocampus, that is the importance for spatial learning and also the recognition memory and working memory. This is a paper from Dr. Kiran Tapalia. He was presented here last year, so many of you maybe saw this data previously before publication. So what Kiran was looking was two specific regions, the subcolon head and pre-subcolon head. And if you look here for MCFS and long COVID patient, we have this increase in the volume for both areas. And when we compare both cohorts with health controls, we have a significant difference. This region of the hippocampus is large in both M and long COVID. And here is again another find we have about this overlap, because both is increased, but both have a similarity. No difference when we compare this part of the brain from both cohorts. Kieran also was looking, this time using the 3T MRI. That's more conventional, is usually what people use for brain neurochemicals. And in this one, he was studying the posterior cingulate cortex. And this paper has many finds, but I selected one that's about the glutamate. We have some talks, I think it was yesterday, about this important part here. And because this I select, so the glutamate level in long COVID and M patients in both was elevated. And you can see here these two groups, the red and the green one. And we have, again, no difference between these groups. And what is important is that the glutamate release, transmission, and metabolism could be affected, what can explain the increase also of the inflammatory cytokines that we have been seeing in different research of M and long COVID. So, okay, I have time. I would like just to present some ongoing research we are doing on NCND. So first, about our IO channels. We are currently doing, we are in the final review of a paper to submit about large cohorts of M and long COVID looking treatment-free, because we want to show and combine it also with data from other group in Australia that did the same to validate this method. So they didn't find similarities with our results, so it's a completely different setup. It's another person realizing then performing the experiment. And we also focusing on other kind of trip channels. We have in these trip channels a family of 28 members with different expressions and with different roles. So we want to do the research focusing other, because as you know, M patients and long COVID too, sometimes one treatment works for one patient but doesn't for other. The same about symptoms, it's too complex and too different. So we want to see how the other trip channels is also in other cells, and to see other potential drugs we can use. We have a PhD student that is completing this year her study, and she's looking the trip channels regulation, but intracellular and intraorganelles, and the calcium mobilization. And this technique, patch clamp, is really complex. It needs a long time of training. So it's something focused on research, no for a diagnostic. So we are looking rapid, a way to identify a rapid identification of this IO channel to be practical, to be applied in different labs around the world. About MRI, our focus is mainly in the connectivity. And we are also starting looking about the machine learning for improve this find and have a short way to demonstrate these results. And the last part is our investigation with other drugs, not just LDN, and the clinical trial we are running. So we are in the middle of the long COVID-1. And we are start soon the MCFS1. And in the MCFS, we will look at the trip channels to see how the trip channels are. And maybe the most important, I would like to invite all of you to go to Australia in November. Our team is organizing again a conference. And like this beautiful place, you can also enjoy. We are accepting abstracts until November. Happy to discuss if anyone wants further information, and please contact NCND if you want any collaboration, conferences, or if you want to just discuss our research. And thank you for your attention. Thank you for our collaborators, especially for Jean, Professor James Baranyuk. Thank you. This is part of our neuroimage team, our PhD candidates, our director, and here our team. So thanks so much for your attention, and I'm happy to answer questions, to have a lunch together, to discuss after. I will have my night here, just return to Australia tomorrow, so if anyone here and want to discuss, I'm here for this. Thanks so much.
Thank you.